DNA replication and mutation. DNA replication is the process by which DNA makes a copy of itself during cell division to ensure continuity of its species. DNA, or deoxyribonucleic acid, is the hereditary material in humans and all other organisms which, when replicated exactly, ensures continuity of its species. DNA is a double helix shape which contains two strands called polynucleotides. The information in DNA is stored as a code made up of four chemical bases: adenine, guanine, cytosine, and thymine, which are known as DNA bases. DNA bases pair up with each other, A with T and C with G, to form units called base pairs. Human DNA consists of about 3 billion bases, and more than 99% of those bases are all the same. The order or sequence of these bases determines the information available for building and maintaining an organism. Each base is also attached to a sugar molecule and a phosphate molecule. Together, a base, sugar, and a phosphate are called a nucleotide. DNA replication is a semi-conservative process, which means that by each strand in the DNA, the double helix acts as a template for the synthesis of a new complementary strand. Synthesis starts with the relieving of tension of the wound bases by the enzyme topoisomerase, as seen on screen. Topoisomerase 1 does this by cutting one strand, while topoisomerase 2 cuts both strands. This model depicts topoisomerase 2, which cuts both strands. Next, the enzyme helicase separates the two strands by breaking the hydrogen bonds between the DNA bases, as will be seen in the model. Single-strand binding proteins then bind to each strand as portrayed by the white spheres, which ensure the strands stay separated. The separation point is called the replication fork, and the two strands formed are the leading and lagging strands. The leading strand goes in a 5' to 3' direction. The synthesis process is started off by the DNA primase, which places RNA bases, called a primer. DNA polymerase then uses the primer as a starting point and adds DNA bases to continue off the leading strand. DNA polymerase does this in a smooth, continuous 5' to 3' direction. The lagging strand, however, is not as simple as the leading strand due to its 3' to 5' direction. DNA primase adds multiple RNA bases, or a primer, which starts the synthesis process. DNA polymerase then adds DNA bases, which join to the RNA bases on both sides. These are known as Okazaki fragments. The DNA polymerase, however, places the DNA bases in the 5' to 3' direction, which is opposite to the direction of the lagging strand, which is 3' to 5'. This is why the lagging strand takes a lot longer than the leading strand. The enzyme exonuclease then removes all RNA bases from the lagging strand. Another DNA polymerase enzyme then fills in the blank by adding more DNA bases, with its complementary base pairing. Finally, the enzyme DNA ligase seals and joins Okazaki fragments and the DNA bases together on both leading and lagging strands, forming one unified strand. Some DNA mutations occur are substitution, in which the DNA bases switch, insertion, in which an extra DNA base is added, which disrupts the rest of the DNA sequence due to a mismatch in DNA base pairs, and deletion, in which a DNA base is removed, which also disrupts the DNA base sequence. However, there is a repair mechanism. Mismatch repair enzyme finds a mismatch in the DNA and removes the base pair, as can be seen on screen. Then, a DNA polymerase enzyme adds the correct corresponding base pair to the DNA base. However, in some cases, not all mismatches can be found, which results in a mutation such as cancer cells.